In this third part of the Pathology AI presentation, we talk about regulatory affairs and the business case. We introduce the regulatory landscape, which can be quite complex, reaching from a central lab model under CLIA or CAP regulations to a medical device development under FDA regulations. We then discuss how rich information data for tissue can be the key to considerably simplify the regulatory pathway. Pathology AI can be provided as a service under a central lab model or as a medical device. Pathology AI is software only, using commercial off-the-shelf hardware. As different labs use different slide scanners already, and those slide scanners account for the major investment in digital pathology, any pathology AI system needs to be slide scanner agnostic and work with any slide scanner that can be found in the clinical labs. As in radiology, pathology now needs to be able to plug and play with different modalities and devices. FDA's preferred complete system approach for digital pathology is no longer financially viable. Pathology AI systems need to be their own medical software devices with a clear and easy path to use commodity slide scanners. A clinical lab can provide lab-developed tests, LDTs, under CLIA or CAP regulations. A LDT does not require the use of medical devices. Actually, third-party software without any regulatory requirements, like Photoshop, can be used. FDA now wants to regulate those lab-specific tests as well, and may require labs to add design controls to the development of the LDTs, not the software, and regulatory approvals. To take pathology AI software and manufacture it as a medical device for distribution into the clinical market, a company needs to implement a quality management system, QMS, for medical device software based on ISO 13485, ISO 14971, IEC 62304, and FDA QSR, and standard operating procedures, SOPs, for good clinical practices, GCP, for clinical trials. Pathology AI systems can move seamlessly from research to a clinical lab to become a medical device. Pathology AI applications can be developed as a prototype until they show clinical utility in an exploratory setting. Then the prototype can be brought into the clinical lab as a LDT under CLIA or CAP regulations to be used in clinical trials and then be distributed under a central lab model. Building a medical device requires to have a medical device software development team and a regulatory and QA team. At this moment, it becomes important to control the software, having done in-house software development opposed to using third-party tools. Using a validated prototype as a basis for medical device development simplifies and de-risks the development lifecycle considerably, which at that point can become a simple waterfall model. Being able to manufacture a medical device that provides the same data as its prototype during a clinical trial opens the door for parallel product development, where a simple bridge study can be used to allow the medical device to use the study data from the clinical trial for its regulatory approval. Rich information data for tissue may be the key to simplify the regulatory hurdles considerably. In a traditional scenario, every tissue, stain, clinical outcome combination requires a different time-consuming and complex FDA approval. Basically, every diagnosis, prognostics, and companion diagnostics needs its own complex FDA approval. Taking rich information data for tissue, which are only measurements that are unrelated to clinical outcome, may only require a simple FDA clearance per tissue type. During drug development, or when using big data in a clinical setting, we would just need to develop scoring schemes that link the already FDA-cleared rich information data to clinical outcome. Much faster, less complex. Flagship is on top of the regulatory requirements and pushing the boundaries. In our previously CLIA, now CAP certified lab, we have established a quality system that deals appropriately with digital pathology and computational tissue analysis. We have also implemented FDA's design controls for LDTs and are in the process of submitting our first 510(k) for FDA-controlled LDT to the FDA. We are ready to take our Pathology AI software and build medical software devices. We have established a QMS for medical device software development and SOPs for GCP for clinical trials. More details on the regulations that apply to digital pathology can be found in a featured article, Digital Pathology, a Regulatory Overview, that I wrote back in 2011 for Lab Medicine. As part of the business case for Pathology AI, we talk about the market, the required competencies of companies that want to play in this space, and discuss what the killer app and viable business model would look like. Let's take a look at some numbers to get a better feel for a business case for Pathology AI. Even though the market numbers are back from 2008, the fundamentals are still the same. The US anatomic pathology market size in 2008 was estimated at $30.2 billion, with an annual growth between 7 and 10%. The market is segmented by subspecialties, which basically correspond to different tissue types. Cervical cancer screening with 26% of the market, prostate cancer with 22%, breast cancer with 8%, etc. Let's use breast cancer as a representative market segment, as it holds 8% of a market that is segmented into about 13 major market segments. In 2008, there have been about 184,000 new cancer cases, and a total of 1.3 million biopsies were taken with an average of 3 cores. With an average reimbursement of $150 per core, including special stains, the market size for the subspecialty of breast cancer was estimated at $589 million. With each biopsy, different tests can be performed, which basically corresponds to different stains. Typical for breast cancer are H&E staining for diagnosis and IHC stains HER2, ER and PR to determine the best treatment option. The average reimbursement of $150 per core is attributed to a mix of different CPT codes with different reimbursements. Assuming an average reimbursement of $100, which is at the low end, 1.5 tests per core or 4.5 tests per biopsy would be performed. Applying 2 days additional reimbursement of $7.56 for computer assistance based on the difference of CPT code 88361 at $141.44 and CPT code 88360 at $133.88 to the 4.5 tests per biopsy, we get an estimated market size for all computer-assisted breast cancer tests of $45 million. Assuming that there are 4 different stain-specific tests for breast cancer, the Pathology AI market in the US for any tissue stain-specific test can be estimated at $11 million. Assuming that breast cancer is 8% of the overall market size, the Pathology AI market in the US can be estimated at $557 million. But it seems unlikely that CMS is going to just add $500 million to their reimbursements. Implementing Pathology AI requires to adapt digital pathology, which by itself doesn't have an obvious business case. Depending on if a lab already has a scanner or not, the additional reimbursement for computer assistance may have to fund the purchase of a digital pathology equipment as well. Assuming that each of the 6,000 histology labs in the US requires at least a scanner that costs $200,000 and has a life cycle of 5 years, the market size just got reduced by half. A quick comparison to radiology. In 2005, R2 Technologies provided a mammography CAT system with FDA clearance, based on a reimbursement of about $25 per test. But just for the analysis, as the adoption of digital radiology financed itself by being cheaper. The market in the US was estimated at $100 million. R2 was able to capture 45% of that market with $45 million in revenue in 2006 when they got acquired by Hologic for $220 million. An estimated market size of 5 to 10 million and the dependence on digital pathology may not allow to develop a viable business case for pathology AI. A central lab model where the test is provided as a lab-developed test LDT under CLIA regulations, or even as a FDA-controlled LDT, would lower the development costs and eliminate the dependence on digital pathology. In the past, image analysis applications for HER2, ER, and PR have been developed by digital pathology manufacturers, but with a completely different business case in mind: to be able to market their digital pathology equipment into the clinical market. Pathology AI is dependent on the adoption of digital pathology, but it will most likely be the driver for its adoption, as it allows to add financial benefits to digital pathology. What is a killer app? What is the right business model? It seems very unlikely that applications that provide the same results as pathologists using a microscope, just providing better consistency or saving the pathologist time, will make a difference. We have seen that very clearly with the computer-assisted IHC HER2 test for breast cancer, the poster child for these kinds of applications. The adoption started very strong between 1998 and 2002, when the additional reimbursement was very high, about an additional $170 per test. By 2002, about 450 ACES systems were placed. The reimbursement changed in 2003, and the additional reimbursement went down to under $60 per test. In 2007, 5 years later, only 250 ACES systems were still placed. Today, the additional reimbursement is under $10 per test. Rare event detections, like pathogens, like acid-fast bacillus AFB, or cellular patterns, like mitotic figures, that could save pathologists a lot of time, didn't even get that far. After all, the gold standard is a pathologist using a microscope. Why change to lose money? Ultimately, it will be the first microscope-impossible test that will drive the adoption of digital pathology. Prescribed cancer treatments are effective in only 25% of cancer patients. Inefficient, expensive, and detrimental to patient health. Continuing with our example, the drug Herceptin acts on HER2 new receptors. In normal cells, HER2 controls aspects of cell growth and division. When activated in cancer cells, HER2 accelerates tumor formation. About 20 to 30% of breast cancers overexpress HER2. Those patients may be candidate for the drug. Herceptin costs about $70,000 for a full course of treatment. One of the significant complications of Herceptin is its effect on the heart. Herceptin is associated with cardiac dysfunction in 2 to 7% of cases. Adverse drug reactions in the US annually account for 100,000 patient deaths, 10 billion healthcare costs, fourth leading cause of mortality, and between 1997 and 2004, 19 drugs were removed from the market due to adverse drug reactions. Pharmacogenomics allows to identify patients who are more likely to respond to particular therapies and who require dose modifications. Stratification of clinical trials, even retrospective, boosts efficacy and eliminates toxicity. Tissue image analysis based companion diagnostics only makes sense when a microscope-impossible test is needed to achieve the required performance for drug approval. As a risk reduction for the performance of a manual microscopy-based test, tissue image analysis can be used on a parallel path during drug development, to be brought in without delay when needed. Companion diagnostics requires a costly medical device development specific for a certain drug. As pharma companies are competing with similar drugs and indications become narrow, the market size shrinks even further. A key problem is a global distribution of those tests. Prognostic tests can be offered under a central lab model, therefore with lower development costs, no dependency on digital pathology, and for a much higher price. But the lab will have to make considerable investments to establish clinical utility and market their products. As an example, Genomic Health has marketed its Oncotype DX breast cancer test based on a quantification of 21 genes linked to the likelihood of breast cancer recurrence in women and the magnitude of benefit from certain types of chemotherapy and hormonal therapy for $3,460 to oncologists through a direct sales force of 80 reps. A prognostic test at $3,000 that could be used for all 184,000 new breast cancer cases represents an estimated market size of $553 million. The true opportunity for Pathology AI is personalized medicine with big data, meaning that we can run standardized panels with multiple markers that provide rich information data for tissue on every patient. This will allow us to make treatment decisions that include the full spectrum of available and new drugs with a single standardized test, and to be able to use this live rich information database to develop new prognostics in the field, correlate patients with existing and emerging health issues, and get new insights. Being able to better characterize our patient population will allow us to circle back to accelerate new drug developments with a smarter patient selection and to provide easy companion diagnostics that are based on those already existing standardized tests. The current reimbursement model does not provide a viable business case for Pathology AI. Pharma companies who want to bring their drugs to market and payers who want to improve patient care and lower healthcare costs need to provide a viable business model that is based on the value that is provided by pharmacogenomics and big data. Continuing with our example, an IHC HER2 test that identifies the 20 to 30% of patients that benefit from a $70,000 treatment and eliminates the risk of cardiac dysfunction in 2 to 7% of the 70 to 80% of patients that would not benefit from the treatment anyhow would save the healthcare system about $50,000 per test. Here is a US-centric view of the relevant competencies for the companies in the Pathology AI space, presented as a spider plot. We are looking here clockwise, starting from the top at technology, science, market, distribution, regulatory, and products. Without going through all the details, we can see in orange the competencies of a traditional single-site histology lab with pathologists providing lab services under the CLIA cap regulations and serving the research and discovery, clinical trials, and reference lab markets. We saw many histology labs adding very simple IHC image analysis capabilities to their menu based on third-party image analysis tools, shown in light blue. But the problem with third-party image analysis and machine learning tools, shown in green, is that they have been designed by technology experts only and are very difficult or impossible to use by ordinary pathologists and biologists, and don't address or lack the real needs in the market. Actually, the key to success in this space is to be able to bring the expertise of pathologists, biologists, image analysis and machine learning experts, and the histology lab together to build the right science and technology, but also to deliver it as a service, eliminating the complexity for the user until specific applications can be provided as a medical device to the clinical market, at which point most of the complexity is locked down and an easy-to-use user interface can be provided. Let's take a look at how Pathology AI companies have to interact with other companies, depending on the product they want to offer. Companion diagnostic products are driven by pharma companies who need a continuous path from their clinical trials to a product in the clinical market. Pharma companies typically provide the data for the development and conduct the clinical trials required for the regulatory approvals. Companion diagnostic products typically involve a partnership with IVD manufacturers who provide the assays and have the appropriate distribution channels. Computer-aided diagnostics can be developed by any technology company. No IVD manufacturer need to be involved, as common H&E staining is used. The key is to get access to the data for the development and clinical trials. which typically implies partnerships with academic medical centers. The technology company is responsible for the entire product management, including R&D, manufacturing, clinical trials, regulatory approvals, sales, marketing, distribution, and servicing. Prognostics can be provided as a service under the central lab model. Tests can be built using third-party tools. The key to big data is the access to the data, the patients, which in the US is mostly in the hands of academic medical centers and reference labs with large footprints. Ultimately, the payers are going to play a vital role in healthcare big data, as it has tremendous value in improving healthcare and lowering costs, but also requires a new viable business model for the institutions and companies involved. To illustrate the different concepts and ideas in the context of familiar applications, we used examples based on well-known breast cancer IHC markers for HER2, ER, and PR. Here's now a more relevant example for the use of pathology AI, where we demonstrate the use of machine learning in an immuno-oncology application that is part of our standard Action.io product offering. We want to illustrate the use of our pathology AI system for immuno-oncology, an application that is too complex for pathologists who are just using a microscope, but when provided with the right tools, pathologists can provide high-quality, high-performance complex testing. Here we are looking at a non-small cell lung carcinoma with IHC staining, CD8 in green and PD-L1 in red. A key component of the analysis is the use of machine learning for the proper classification of tumor cells versus cells in the tumor microenvironment. The pathologists provide their unique expertise in a convenient way by providing example regions for the different tissue types to then train the system on the fly. Here we outline one region with tumor cells and two regions with cells in the tumor microenvironment. Using just these regions, we will achieve an estimated accuracy for the cell classification across the entire slide of 84%. The system provides the results quickly, in the order of seconds. The system displays all detected cells, here over 1 million cells for the whole slide with different colors for the different tissue types. Here we display tumor cells in red and the cells in the tumor microenvironment in green. When there are cells that have not been classified properly, then the regions with those misclassified cells are just added to the training. This is an interactive and iterative workflow until the cells on the whole slide have been classified properly. This way, the expertise of a pathologist is used in the most effective way for each patient, providing the highest quality of data. By adding just these example regions, our estimated accuracy for the cell classification across the entire slide goes up to 92%. To save pathologists time, the initial training of the system can be performed by a trained analyst. Again, the system provides the results in the order of seconds. The key problem in pathology, the variations between different patient types, is eliminated by creating a different classifier per patient type. Different patient types and their characteristics are identified automatically in this workflow. No pre-training of the system is required. No bias from training data exists. Proper controls are provided by the pathologist, who verify the proper detection and the proper classification of the cells. A key concept of our pathology AI system is that it provides high-quality, rich information data for tissue, which means tissue type specific cell distributions with over 1,000 biofeatures. Think flow cytometry data, but with tissue context. Rich information data for tissue enables healthcare big data for pathology, allowing us to benefit from the power of data science. The measuring of features and counting of cells are best performed by a computer and can be verified properly as part of software verification. The importance of whole slide analysis becomes clear when looking at the heterogeneity across the slide. But whole slide analysis does not mean to analyze the whole slide. The pathologists need to identify the right region for the analysis, the tumor with its tumor microenvironment. Here we discard 20% of all cells. Our biofeatures include the quantification of the stains. Here we show CD8 in the tumor microenvironment. Now PDL1, and here CD8 in the tumor. Now PDL1. The important biofeatures can be summarized in form of scorecards. These immunograms are part of our standard Action.io product offering. Pathology AI means to provide the pathologist with the right tools. To replace the microscope, not the pathologist. The true barrier of pathology AI is the business model, not the technology. We have developed our pathology AI system over the last 8 years, and it has been working in our lab, meeting the needs of the market. To get pathology AI into the clinical labs, payers need to provide a value-based model that creates a viable business case. The adoption of pathology AI, and therefore digital pathology in general, will be driven by microscope impossible test that requires a pathologist to use pathology AI, similar to the Immuno-Oncology demo we just showed. Pathology AI can be seen as a new modality that provides rich information data for tissue. Rich information data enables healthcare big data for pathology, which is a key to improve healthcare and lowering healthcare costs.